Well, we're a team that is purpose-driven, and that purpose is to save and improve millions of lives through a singular focus on local patients' needs. And it's really because of these local patients' needs that we are with you today. I think patients' needs today are a little bit special, especially in certain parts of the world. I guess the country I'm from is no exception. Well, we're a little bit early in that disease, but for sure we need to take care. Otherwise, we could have the same fate of countries that had infections by the tens of thousands a day. I think these are the numbers from yesterday, and the numbers, as we can see, are escalating quite fast. Well, we can all know that the symptoms are, well, most of people don't get symptoms but are contagious, and then maybe 20% are between mild, severe, and critical. There are different products that are being repurposed, and we as a company, in addition to the collaborations we have with different companies, including a company in California called Gilead Sciences, we're working very diligently on finding a real cure and enabling that cure to be in the markets where we're present. Well, as you can see, there are different molecules that are being repurposed because nobody really has time to invent something new because that takes years. And as you can see, especially in the inner circle of phase 3 trials, there are different principles that are being tried. Remdesivir, favipiravir, as well as others are leading antivirals that the world is working on. We are dedicating most of our resources to combat. Well, we're a company that does ophthalmic solutions. We do diabetes medication, but we are very much an antiviral or a company that is focused on combating viruses. We see that we are in a state of war. The only difference is that the enemy is common. The enemy is common for all countries in the world. We see that the armies wear white robes. These armies are healthcare professionals led by doctors, nurses, pharmacists, and other. And we see that we need to proceed in this war with everything we have. And really, in a war situation, nothing is left, nothing is delayed, and this is why we are here today. I think there is no better time for us to get together. I think there is no better time for us to engage with the aim of collaborating, to hold the line and try to defend our patients from this attack. I think a lot of people would be exposed to this virus, but it's not the only problem. Of course, other diseases that may be as important as, but a little bit more chronic, need us to work 24/7 to make sure that these patients are taken care of. Well, in terms of critical care, this is a press release that we've had in Arabic, and this was directed towards Egyptian healthcare professionals, basically announcing that we have joined a global consortium to make available tens of thousands of small, simple, yet very effective ventilators available for Egyptian as well as North African patients. If you allow me maybe another minute or two, I can go through quickly to try to explain the type of organization that is trying to go into this war. Well, we're an organization that has a long history. As you can see, we've had a distribution company over 103 years ago, as well as the second manufacturing facility in Egypt. Back then, the language of science was French, so this company was called Les Laboratoires Delta, which was the second manufacturing facility in Egypt and was able to register important products, including this product that is still available in Egypt by the name of Urosolvine. But unfortunately, Egypt, as well as other countries, became socialist in the 60s, and these two companies were nationalized. Our future, well, we want to be a significant company. We want to be a company that has economies of scale. And to do that, we think we need to be one of the top 20 specialty companies globally, but still headquartered in a home market in Egypt. These are the countries we're in, over 40 countries. And we were proud to have the first Egyptian products in Canada, as well as have a European base starting 2018, was able to register many products famous in Egypt in countries like Germany. We operate 3 facilities, and we are using these 3 facilities in the current war against Corona, as well as against other diseases that we are trying to hold the line and stand for. Well, this facility in Cairo is a facility that is approved by the EU Drug Regulatory Authority for the third time. 2014, 2017, 2020. This year we're able to reapprove the facilities, and we're able to add sterile lines, including ophthalmic solutions, suspensions, and other sterile products, including vials, which hopefully soon will start to manufacture antivirals, including the ones that are used for the coronavirus. Behind these facilities is a very strong quality assurance and quality control system. Well, this is a picture of the entry of the sterile line for vials and ampoules. We have different products, different lines in this facility. The second facility that I want to talk about today is the biggest investment in North Africa, a facility that was started in 2017, and now we would like to have this facility approved by the FDA. Last year, we were able to go successfully in a mock audit, and this year we're expecting the FDA to come and approve such a facility. This facility has many advanced lines, including the cephalosporin line that is able to do joint filling of 2 ingredients into these vials. Well, I don't want to go on for too long. This is our rank in Egypt as a manufacturer. The other element that I wanted to focus on is the element of research and data analytics. This is our standalone R&D center, was inaugurated end of 2018 with this luminary Egyptian scientist, Professor Dr. Sir Magdi Yaqub, as well as we had the honor of having the likes of Professor Dr. Mustafa Sayyid. Well, the most important part of the center are the scientists that are in the center, and these are the scientists that are enabling us to locally manufacture active pharmaceutical ingredients that will be used in the fight against corona. The last slide I will use with you this evening is a slide where we promise that 100% of our products are completely serialized, which is in full compliance with the EU directive that came out into full effect in February 2019. Basically, every package of product has this unique serial number, like a product ID, but it's not for the product or for the batch, it's actually for every box in every batch, for every product, for every manufacturing site. With this, I would like to thank you. We're honored to be hosting this meeting, and we would like to thank Professor Armia very, very much for bringing all these people together, because again, there is no better time for us to engage. There is no better time for us to collaborate than now. Thank you all.
Thank you, Dr. Riyad, for your speech. Thank you so much. We are going to have our meeting. I'm going to start by introducing the meeting. I am Dr. Ashraf from Cairo, Egypt. I am really honored today to moderate this event, which is a live streaming from Cairo, Egypt. It's an international ophthalmology meeting about new approaches in keratoconus management, which is organized by Eva Pharma, Egyptian company. Nothing can stop us conducting science, even in the era of COVID-19 pandemic and our quarantine at home. Continuous medical education must be continued, even if all the big international meetings have been canceled. It's my extreme pleasure today to have my dearest eminent ophthalmology friends from all over the world, Dr. Cosimo Mazzotto from Italy, Dr. Miguel Richichi from Italy, and my dearest friend, Dr. Eric Martins from Belgium. We are now live for all ophthalmologists all over the world from Cairo, Egypt. Thanks again for the Eva for supporting this continuous medical education. So we are going to start by Professor Cosimo. He's going to talk about what's new in the crossing. He promised me that he's going to talk about a very new topics that maybe didn't talk about it before. It's coming future for the cross-linking. So the mic with you, Professor Cosimo.
Thank you. Thank you, Ashraf, for this kind invitation. Can you see my screen now? My slides?
Yes. Yes.
Okay,
here.
perfect. Thank you. First of all, let me say Happy Easter to all the audience. Thank you to all of you. This is a particular moment for each of us. And it is a great pleasure for me to be in this meeting with you. I will speak about the future of cross-linking. The question is if the future of cross-linking will be a beyond. I hope to answer to this during the presentation at the end. As you can see here, in the last decade, there were a lot of innovations in corneal cross-linking. We started from Dresden Protocol, and we go to the EPI accelerated protocols. We had also the pulsed light accelerated protocols in cross-linking. We have recently standardized the nomograms for treating thin corneas. Myself, nomogram and the new sub 400 protocol. You know also that we had new enhanced fluence pulsed light iontophoresis protocols, and also the boost oxygen EPION protocol, together with the novelty of the slit lamp cross-linking. What was known and what we recently added to this topic, we know that when we work with epithelium in situ, its antioxidant systems are radical absorbers, reduce the cross-linking amount, reducing oxygen availability, and attenuating the consistently a part of UV light energy of fluence. Increased UV power irradiance in the accelerated protocols without changing the energy and the exposure time and compensating the oxygen consumption shorten the treatment without increasing the stiffening of the corneal stroma. So you know that the Bunsen-Roscoe's law of reciprocity doesn't apply completely to the photochemical pathways of cross-linking. Another problem was the demarcation line after cross-linking showed that UV, riboflavin, and oxygen interaction were correct. And most recently, I have published a major review together with Gregor Wallensack, Ebersberg, and Friedrich Reisbuch from Dresden, that we know that there is a saturation level of cross-linking. So the power of cross-linking can grow not only horizontally, but also into the depth of the cornea. The science behind EPION. So you know that we need the adequate homogeneous stromal concentration of riboflavin. We need adequate transmission of UV fluence. We need adequate oxygen diffusion and availability, driving sufficient cross-links formations. And we also need adequate penetration depth of treatment, ensuring sufficient cross-linking density and saturation effect. You know, if an inadequate amount of any of these four components induce an inadequate over cross-linking biological efficiency. Each of us know the advantage to have an EPION efficacious cross-linking because of its low invasiveness, because of the less complications, because of the more comfort, reducing the risk of infectious keratitis, post-operative stromal wound-related complications such as AIDS, the reduction of pain and the nerves damage, and to have also a faster visual rehabilitation and recovery. But each of us know the limited efficacy of the old protocols compared with epithelium off. We know also the long-term ectasia instability and the less functional outcomes of the chemically enhanced old protocols. So the limits were the 30–50% reduced bioavailability of 0.1% riboflavin in the stroma. Another important limit was that we used the same energy, the same fluence used with and without epithelium, the 5.4 joule. We don't understand the importance of the oxygen consumption when we work with epithelium on. and the lower intraoperative oxygen infusion. Everybody of us know the unevenly detectable demarcation line was about 30% after Epion, and the less corneal flattening and high-order aberrations reduction in the old Epion, and also the mid to long-term ectasia instability, especially in pediatric patients, and also the high retreatment rate that in literature is published from 50% to 100% after 2 years from the first Epion treatment. So this is what I called the 4-level compensation of the new Epion era. First, we increased the riboflavin concentration from the 0.1 to the 0.25 according to high precision liquid chromatography studies. We increased the UVA fluence by 30% from 5.4 to 7.2 joule and up to 15 joule in the photorefractive intrastromal cross-linking boost oxygen treatments. We use the pulsed light 1 second on off to increase intraoperative oxygen diffusion. And 4, we use supplemental oxygen via boost goggles to boost up oxygen diffusion. This is the work that demonstrated in literature the concentration of riboflavin to the stroma. And you can see that if we use iontophoresis loading, we have the halved dose and the bioavailability of riboflavin into the stroma compared to passive diffusion of the EPOF treatments. And if we use chemically enhanced solution, we have 1/3 of concentration. So this was the first level of compensation, the increase of the concentration of riboflavin from 0.1 to 0.25. This is an important work from Lajos Kozlovari, showing that when we do a cross-linking, we are working in a waveband of 370 nanometers. Sorry. So when we work with 370 nanometer, the UVA absorption by the antioxidant systems of the epithelium is about 30%. So, and another important is that it is necessary to wash completely the biofilm over the corneal surface because we need to eliminate completely the riboflavin shield that could be another limiting factor. So, according to this study of photobiology, we need to increase the 30% of fluence if we want to overcome the epithelial barrier and the photoattenuation by antioxidant system of epithelium. So we go from 5.4 to 7.2. Look at this very interesting paper of the Journal of Physiology of 1972 that explained very well the oxygen consumption levels layer by layer. And this paper, this study evidenced that epithelium consumption is 40% of oxygen during the treatment. So the third level of compensation was the introduction of the pulsed light treatment oxygenation. This is the photochemical kinetics of corneal cross-linking. You know, we shift from type 2 to type 1 reaction when we start the UV, and the oxygen is consumed in a few seconds. So the oxygen compensation with pulsed light was done. And it takes a long time to replenishment. It takes about 4 minutes to fully replenish in atmospheric conditions. So if we consider these dynamics, we strongly we can create an hyperoxic environment by using supplemental oxygen intraoperatively, increasing linearly the oxygen concentration over the ocular surface. And we know also the Pulsed light from our articles demonstrated that the advantage of the increased depth of the demarcation line, we improved stromal oxygenation and the overall cross-linking efficiency, and we lose also less microstructural damage. This is a comparative view of different instruments, corneal OCT, shine through camera, and in vivo confocal microscopy, showing that if we use a pulsed light, we go more deeper in demarcation line depth. Also, the first authors of iontophoresis protocol recognized that oxygen diffusion may limit the biomechanical effectiveness of the old original iontophoresis protocol. So what I did, I changed the protocol of iontophoresis. I changed the protocol, and this is published in Journal of Refractive Surgery, because I introduced, first of all, the change of fluence, going from 5.4 to 7 joule, increasing by 30%, and also adding the pulsed light to the treatment. So I choose the 18 milliwatt UV power with 7 joule of fluence, pulsed one on, one off, and it was surprising that we finally see the demarcation line in over 80% of treated patients at first month postoperative, in a depth of, in average depth of 285 microns, plus minus 20. It was impressive, this result, only changing 2 parameters, the fluence and the pulsed light to Epion treatment, because iontophoresis is only a loading system for the riboflavin, but we changed the irradiation parameters and the oxygenation parameters. So finally, seeing the demarcation line at about 300 microns of depth. These are the results that were amazing, and the K-mass flattening show a statistically significant improvement with a minus 1.5 diopters that was very close to Epi-off treatment. This is the last paper that was now accepted 15 days ago in the Journal of Refractive Surgery with the 3 years follow-up of the new iontophoresis protocol, demonstrating very amazing results. So the demarcation line was seen over 80%. The treatment stabilized ectasia progression in the mid to long-term follow-up of 3 years. These treatments are suitable for an outpatient cross-linking procedures. So in this case, no supplemental oxygen is required. You can do also bilateral procedures, and this protocol is also included in the slit lamp CI device cross-linking machine. This is the setup of the Epion protocol that I believe this is the most efficacious Epi-on protocol actually in our clinical practice, beyond the mechanism of riboflavin loading that you want to use if chemically enhanced solutions or iontophoresis. But let's go to the 4th level of compensation. So if we have a controlled oxygen compensation, we increase in 90% of saturation, increasing from 21 room air oxygenation to hyperoxic environment. We increase the diffusion rate of oxygen during the treatment by 5 pair. So the 4th level of compensation can be reached by intraoperative supplement oxygen. And as you can see, the level of oxygen, if we introduce supplemental intraoperative oxygen via boost goggles, is maintained during the overall treatment irradiation phase. Take a look at this study that showed that biomechanical efficiency of Epi-on with hyperoxic environment is very similar to Epi-off. So I show for the first time at international level our first European study on photorefractive boost oxygen Epi-on cross-linking. This is the demographic. We treated 24 patients, 27 eyes. 3 patients were treated bilaterally with a bilateral simultaneous treatment. This is the mosaic system of Avidro, the KXL2. This is the system. This is a probe to check the oxygen saturation inside the mask. This is the screen with the eye tracking, active eye tracking system. And this is the boost goggle that are connected via a tubing to humidifier and to the oxygen delivery system. So take a look to this image. We had 2 demarcation lines, to have double demarcation line. The first demarcation line was for the 7.2 joule fluence, and the deeper demarcation line was the 10 joule fluence. This is the first. This is the second demarcation line. Take a look to this OCT opto-view image. The first line and the second line, because we use 2 different fluences and different exposure times in this kind of photorefractive intrastromal boost oxygen. Take a look to this picture. It is impressive to see very clearly that we had 2 different demarcation lines according to different fluencies and according to different exposure times. And take a look to the results. We had an impressive corneal flattening at the apex of the cone. This is the differential map between pre and post-operative at the first month of observation. And you can see the refractive equivalent differential map that are very impressive. We had over 4 diopters of flattening. This is another case. Let's show you the flattening in the central area. Another case that was really impressive. You can see the anterior tangential differential map and the refractive equivalent differential map on the apical region of the cone if we use boost oxygen transepithelial photorefractive intrastromal cross-linking. So take a look to the depth. This was my major review on the ocular surface, documenting all the penetration treatments. So you can see the conventional epithelium off-dressing protocol went to 350 microns of depth, and you can see the boost and beyond go to 370 microns on average depth. So who drives the penetration here? The fluence, the pulsed light, the concentration of riboflavin, and the radical formations in the supplemental oxygen via those goggles. You know, this is my nomogram, but I think that Brillouin microscopy may assess the biomechanical efficiency. However, the demarcation line will remain inefficacious and is needed for the evaluation of the safety, penetration, and the performance of any type of cross-linking procedure. So in the conclusion, the demarcation line, finally, with this 4 levels of compensation, are clearly evident in the majority of cases. The boost oxygen induce higher corneal flattening and higher order operation reduction compared with the eye room beyond treatments. The flattening that we obtained are on par with the off, if not better. We have no minimal issue at the first month post-op, and the patient recovery is faster with a reduced glare disability, with less potentiality of AIDS, with no infections. The time to contact lens fitting is shorter, and we can do also bilateral simultaneous treatment, also for prevention, not only for ectasia progression. It is a usable method in outpatient modality, also at street level. Of course, you know that we have short follow-up. The only treatment that actually have 3 years follow-up is the new Untitled protocol with enhanced fluence and pulse light. And we need further developments, of course. We need larger follow-up and cohorts, multicentric studies to establish the long-term efficacy of these treatments. So to answer to the first question, yes, I think the epion is the future of cross-linking because the gap of efficacy between epioff and epion is progressively getting smaller. So thank you for your kind attention.
Thank you so much, Cosimo. It's a very interesting and high standard presentation, really. And the epion in the future for cross-linking, as you said, this is true. You have some questions. Can I share it with you?
Okay.
The first question is, what the possible complication or disadvantage for this procedure?
So we don't have complication. We have also an epithelial suffering. So we have a punctate diffuse epitheliopathy after this treatment. So the post-operative regimen is strictly important. We manage this patient like epioff patients. So we put the soft therapeutic contact lens on, and we use mydriatic drops, and we remove the lens after 24 hours. So we don't have complication. We don't have epithelial growth retardation. So it is a good treatment without any complications in our experience.
Do you do this procedure for people below 18 years of age or above 18?
We don't treat pediatrics actually with this procedure. Only young adults over 18 years.
Do you think that anatophoresis double cycle can help better penetration?
Yes. Yes, actually, I think that for the riboflavin loading, the iontophoresis technique is superior to each chemically enhanced solution.
And what is your regimen for post-operative care?
The regimen is the same of epioff. In my experience, it is better because we had pain in these treatments. So the pain is important because we have the sensitive termination of epithelium. So I think it is good to use a non-steroidal anti-inflammatory drug, also topical and systemic. I use ketorolac drops and ketorolac for systemic therapy. And it is really efficacious to reduce pain after this treatment. So I use antibiotics, lubricants, and there's a soft bandage contact lens for 24 hours.
Do you repeat this procedure or it's not repeatable?
No, it was never repeated. The patients are stable with the new iontophoresis in a 3 years follow-up. And actually with the boost oxygen, we have the first experience of 1 year follow-up for some patients and with an average follow-up of 6 months. And the results are very, very good. And
the healthy of the endothelium, because you are reaching very deep in the cornea, it's not harm for the endothelium.
I don't recommend to use the boost oxygen in corneas actually.
Okay.
We need the 400 microns of stroma to use the boost oxygen actually because we go very deep. And I think we need the nomogram like my nomogram in the epioff treatments accelerated, because if we adjust the fluencies, so we could go from 7.2 fluence to 10 joule, increasing the step by 30% to set our demarcation line depth without changing the saturation of oxygen.
Thank you, Cosmo. You have a lot of questions, but we are going to answer everything at the end because a lot of question answers is coming. So now I'm going to introduce. Thank you so much for your presentation, Cosmo.
Thank you.
Now I am going to introduce my friend Miguel Riquiqui from Italy. He's going to talk about what's treating for surface ablation for refractive keratoconus.
Hello, everybody.
Please, Miguel.
Hello, could you hear me? Well?
Yes, we hear you well. Yes, excellent.
Okay, I would like to thank you all for this kind invite, and also to thank Cosimo, because he introduced brilliantly the theme of cross-linking. The topic of my speaking will be the combined procedure. In this case, laser excimer surgery plus cross-linking. And as you know, this is a very controversial topic because in these years we have a lot of protocols, and I'm going to analyze these protocols and my personal protocol. So why all the people struggle for a refractive effect of cross-linking? Because sometimes the big effort that you do with the cross-linking alone is not so appreciated by the patient, because cross-linking, as you know, has not the wow effect. The cross-linking is a revolution for halting the keratoconus. But usually when the patient is in the late phases and can see very well, he will continue to don't see very well. He will have some improvement, but by the years. So this kind of thing is not so appreciated by the patient. And the other way for struggling for minimal invasive procedure is the fear to be cut, because you know, the patient, if the patient can save a corneal transplant, he's very, very happy. So the first approach to the patient is the minimally invasive procedures like cross-linking or combined procedures. But what is cross-linking plus? Cross-linking plus essentially is not a refractive procedure. We're not going to remove the glasses from the patient. The main target is to first to regularize the ectatic cornea, improving the best spectacle correctable visual acuity, and also contact lens fitting, and mainly to reduce the corneal aberration. So we have never to give the full solution to us and to our linking plus procedure. If you look at this the diagram, you will see how if you have a cornea, an ectasia that is stable, but you have unacceptable visual acuity, you can switch versus a combined procedure. The same thing when you have a progression and when you have an unacceptable VA, visual acuity. Also in this case, you will go for a cross-linking plus procedure. But if you have a patient with a very early or intermediate keratoconus and the visual acuity is acceptable, you just go on cross-linking. You do not need to do an additive procedure. And the other case in which you cannot use this kind of surgery is when your cornea is at a deep scar in a very, very early, very advanced keratoconus. So in this case, you cannot do the miracle, so you go for a corneal transplant. But you have to use the cutting edge technology if you want to approach this kind of surgery. It's not an option. You cannot use older machine to do cross-linking plus. And why? This is one of the first issue that in which we fought a lot of years ago with the Cosimo, the intracorneal shrinking when we was using a solution of riboflavin with a 20% of dextran. This is a hyperosmolar solution that really pump out the water from the cornea, also during the loading phases with the riboflavin. And if you can see, and I will show you with a pointer, you sometimes with all the riboflavin, you start with a cornea that is more than 500 thickness, and only just 30 minutes of the loading phases, you have a cornea that is half, minus 250. That's because just only during the loading phases, the dextran pumps out the water from the cornea, So you start to cross-link a 500 micron cornea, but after 30 minutes, you continue to do the UV light on a cornea that is just 300 micron. So that's the main reason for all the complications like corneal burning, scars, endothelitis, and this kind of things was relatively frequent with the Dresden protocol and the dextran riboflavin. That is the reason why I did this study, also with Cosimo and other colleagues, in which I measured with a new solution of dextran-free riboflavin the thickness with a OCT during the surgery. And what I found is that with a dextran-free riboflavin solution, the cornea is quite the same during the whole procedure. And this is very safe because you start with a cornea and you finish with a cornea with the same thickness. And this is the most important thing for safety, especially if you're doing an excimer laser before. The other reason for which you have to keep out the oldest cross-linking machine is that they do not have a flat, an optimized beam. So with an old machine, you have an hotspot into the center, so you have more than 8.4 joule into the center, but you have a steep decrease if you go versus the periphery. So usually the keratoconus is a peripheric pathology of the cornea. So you have less energy just in the apex of the cone and a lot of energy into the center. That's the reason why you can see a demarcation line that is very deep into the center, but is very superficial into the periphery. The new machine had a top hat optimized beam, so you have an even delivery of energy from the center to the periphery, and you have an even demarcation line. So this is really important to test the efficacy of the procedure. And as Cosimo told in the previous talk, you can adjust the energy of the cross-linking if you look at your single cornea, because there is no one keratoconus that is equal to the other. Each keratoconus is different, so you have to titrate the energy for each cornea. To do to reach a good depth of the demarcation line, also in thin cornea. So you are you are efficacious, and really you are very safe. The first thing is not to harm. You have to regularize the cornea, but you have also to strengthen the cornea. So if you go to ablation more than 50 micron, it will be very, very dangerous. It's not be a therapeutic option, but is a iatrogenic dam. And this is a scheme of the surgery. If you can see, you first perform transepithelial ablation, and when you do this, the epithelial removal has this kind of trend. In the zone of the apex of the cone, where the epithelium is thinner, we will also ablate some stroma. So also, if you just remove epithelium and not do further ablation, you will have some kind of a regularization just in the zone of the apex. And in the zone in which the epithelium is normal, you'll have also some epithelium that will also shield the cross-linking. The other good things of transepithelial ablation is that you can isolate just the zone of the cone, just the apex and just the paracentral cone. So the cross-linking will focus more just on the ectasic portion of the cornea. So you will focus the flattening effect just over the cones. This is the real life. The beautiful of this surgery is that a really no-touch laser surgery. So the patient just see the laser above him. As you can see, you first remove the epithelium and do some kind of corneal regularization. And then, without touching the surface of the cornea, you start with the accelerated cross-linking. And as you can see, the ablation is also decentered on the apex zone. So also the ablation is not centered on the corneal vertex, but is centered on the apex. The other good things of doing surgery transepithelial is that you regularize the epithelium. As you know, the epithelium is thinner. After the transepithelial approach, you will have a more even epithelium thickness also over the cone. So this is a very good thing, because you also not only regularize the stroma, but you also regularize the epithelial map. The other advice and the other improvement that I put in this surgery, as I told you, is to move the ablation toward the apex of the cone, not on the vertex. There are more reasons for this. Central corneal ablation not fully cover the cone area, so you need to increase the ablation diameter. If you center into the corneal vertex, this will induce more ablation and more flattening effect, not on the apex of the cornea. And if you center the ablation on the apex, reduce the volume of the ablated cornea, you will have a more focused flattening just in the cone area, and you have less stromal ablation on the apex. This is a typical example. This is a conventional keratoconus, central because the apex is inside the 3 millimeters. I just moved the ablation over the cone area. And as you can see, I just removed, in these cases, 26 micron. You see the differential map in terms of thickness, but I have more than 8 diopters of flattening, exactly where I want. So this is the pre-op and this is the post-op. This is a completely different cornea. But what is the difference? The difference is not only the visual acuity, but to my opinion, the most important thing is the reduction of the coma, the aberration. So the patient started with this situation from pre-op, correct vision, and now with the spectacle, this is his quality of vision. We have a very strong reduction of the aberration. So the main target is aberration improvement, not the refraction. This is another example. It's the same patient, the 2 eyes. The left eye, because he has a thicker cornea, 484 micron, and 52 diopter of curvature in the apex. For this eye, I chose a topo-guided transpure K plus cross-linking. For the other eye, in which the cornea was thinner and I have a bigger curvature, 56 diopter, I choose just to remove the epithelium to be more conservative. So just epithelium removal with the laser and then cross-linking. And this is the result. This is the first eye in which I performed the customized TransPRK. I always choose a minimized depth protocol just to save more tissue. And as you know, after this kind of protocol, I saved more than 20% of volume, 12% in the volume of ablation, and 18% in the maximum ablation. So I have a maximum ablation of 85 micron epithelium, and the final result is a 61 micron less, but a very big improvement in the coma aberration. This is the pre-op, root mean square of 2.9. This is the post-op, 2.4 of RMS, and this is the difference map. It's a very huge difference in aberration. Okay, this is the other eye, in which the cornea was thinner and I have a bigger curvature. So I just removed the epithelium with the laser, and finally I just have an ablation of 26 micron, and also a very noticeable difference in aberration map. So I was conservative, but I have the same a good result in terms of aberration. This is another example, and this is another kind of corneal ectasia, not keratoconus. This is a pellucid marginal degeneration. And in this case, the study protocol is beautiful because, as you know, in the PMD, you have a good thickness here when in the ears, and the thinnest zone is here. But when you do a customized laser ablation, the ablation will be more here where you have more tissue. And I will show you this patient in which that was refused for driving license because of high astigmatism and low vision, and it was very desperate because his fear was that his wife could carry him for life. So this is the ablation. As you know, I performed an optic zone of 7 millimeters, a ablation of 163 micron with epithelium, so 100 micron maximum of a stromal. And this is the refraction that I put, that is not the real reflection. I pointed at corrected the half of the cylinder, so minus 3.50 diopter. And this is the result before and after for this is a left eye, and the pre-op is here on the left and the post-op is on the right. And I have a big improvement in visual acuity and also a reduction in a cylinder. So this patient is very, very happy. He passed brilliantly the drive exam, and also I have the compliments from the commission. So the STAR-X is for many, but not for all. So when you have this kind of nipple cone, very, very steep, in this case, you have just to remove the epithelium, because if you do some kind of a corneal regularization, you will ablate really a lot of tissue, and you will waste for sure this cornea. So just remove the epithelium, and doing the accelerated cross-linking could also lead you to a very beautiful result. This is the patient after 1 year, and he has a 7 diopter of flattening into the center. This is always a very steep cornea, but this patient can now fit a good contact lens, and he is happy without corneal transplant. The good things of the sterile protocol is that the peripheral cone patient gain more than 2 lines. So this, the group 2, the group of the patient in which the cone is peripheral, start very bad, but finally they earn more line respect to the central cone. So this is the beautiful of this surgery that is particularly fitable for the peripheral cone. The cornea is very stable. All the patient has improvement, and all the patient have a stable posterior elevation map after 2 years. And the great aspect of this surgical approach is not only the quantitative improvement, but the improvement in quality of vision. You can also combine with intracorneal rings and with the cares that was introduced by Susan Jacob. And as you know, the sterics reduce but do not eliminate the need for corneal transplant, that always remain a surgical option. So my only advice is that you have to take care about the patient selection and treatment, because sometimes a good intention will not ever lead to a good result. And I thank you all, and I wish you an happy Easter to all the members. Thank you.
Thank you so much, Miguel. It's a very, very interesting topic. But I just want to ask you, if you don't have the machine for the cross-linking,
yes,
can you do it without this machine?
Okay, there are in the market a lot of fixed energy machine. So usually the fixed energy is 10 milliwatt, 5.4 joule, and 15 milliwatt, always a 5.4 joule. So if you have this machine, you can perform this kind of surgery in the cornea that has at least 450 micron, okay? So do not try to go with a lot of energy in the cornea less than 450 micron. If you have totally adjustable machine, you can go below.
There is an idea that they use the contact lenses, which is not the UV protection, and they make an opening in the contact lens. So the other part of the cornea does not take the cross-linking, and only the open part in the contact lens will go to have the crossing.
Okay, this is really tricky, but there is a good technique that also Cosimo published that he called the epithelial island cross-linking. So sometimes, if you want to protect the thinnest part of the cornea, you just do manual epithelial removal and leave an island of epithelium just in the thinner part. So you can easily cross-linking the other part, saving the thinnest part of the cornea. It's more practical.
We have also a lot of questions. We are going to have it in the end.
Yes.
So we can go to the next presentation. I'm going to introduce my dearest friend, Dr. Eric Merton. He's he's going to talk about a very interesting topic. He's very specialized in this part, the ICL, and how we size the proper size for the ICL. Your talk with you, Dr. Eric. Thank you so much for sharing your topic with us.
Ashraf, thank you very much for the invitation, first of all. I'm very impressed with your webinars. I must say, as president of ACOS Europe, we had to cancel, unfortunately, our conference in Florence in the beginning of June. But things like this, the webinars, and also from ACOS, we will launch on some webinars in the near future as well. So, but thank you very much for giving me the opportunity to give my talk here.
Thank you so much, Eric.
Different. Well, Ashraf, thank you very much. It's my real pleasure. So it's a little bit different than the other speakers, but after keratoconus correction, after cross-linking, you still have some refractive error, and you need to deal with it. So, and one of the most elegant ways to deal with it and to correct refractive error is with ICL implantation. And then we come to the subject of ICL sizing. So, and I've done this for more than 20 years. And what is the most elegant way to size the ICL and to get the most or the best results with these implantations? And for a couple of years now, I'm working with an OCT, anterior segment OCT, and looking at the sclerospur to sclerospur. These are my financial disclosures. I'm a consultant for Star Surgical and from CSO, the company who provides the anterior segment OCT. This is our webinar and our eminent speakers. So the device I'm using is the OCT MS39 from CSO, and it has a lot of capabilities. It does some anterior segment measurements. It gives us high resolution corneal imaging. It does topographic follow-up. Very important, it analyzes dry eyes and tear film quality. You can screen for keratoconus preoperatively. You can plan cataract surgery and IOL calculation. It gives you corneal wavefront analysis, gonioscopy, glaucoma angle measurement, and pupillography. But for this talk, the most important things are anterior chamber measurements and dry eye and tear film analysis. Now, this instrument gives you an automatic acquisition of anterior segment parameters. It is highly repeatable, it's very fast, non-invasive in respect to the UBM machines where you have to touch the eye. And well, in COVID-19, post-COVID-19 area, patients who want to be safe and want to be reassured. And when you can do a measurement without touching the eye, this will create a high level of confidence from the patient. So non-invasive will be very important in the near future, and it is very patient-friendly. Now, how can we make ICL sizing predictable, easy, and also very important, repeatable? How do we do it preoperatively? First of all, when we do the acquisition, we check the acquisition quality. As you can see here, the machine itself gives us the acquisition quality. When you see this, when all the bars are in green, you're very sure that the acquisition was well done and you can rely on the results. The parameters we are checking are the dry eyes, as you can see here. The patient has to blink, open his eyes, and then the machine measures the surface of the cornea where the tear film breaks first. So you can easily see where it breaks first. And here you can see on the graph the NIBT, the non-invasive time, which is very important to talk to the patient. When you're not dealing with it, the result will be less than optimal, even with refractive corneal surgery or with ICL surgery. We can tell the patient, we need to take care of your tear film first before we do any kind of procedures on your eye. What else does this machine do? It gives you a choice of 4 different maps. Here are the maps. And when you can see here, so this button is a very important button. You always have to click it when you do any kind of refractive surgery, even cataract surgery. It gives you the epithelial thickness map. Is the epithelium, is it very the same thickness all over the cornea, which is very important. It also gives you the refraction of that epithelium over the cornea. It gives you the central corneal thickness and the anterior chamber depth as well. So it gives you the anterior chamber depth, the epithelial thickness, and the total anterior chamber depth, including the corneal thickness. We also look at the angle to angle and the anterior chamber width, which means what is the distance between the scleral spurs. And when we look at it, always check first how you measure it. So we measured the scleral spur to scleral spur in the horizontal region. The machine itself gives you already the scleral spur. Where is it? And if you're, as a clinician, you don't agree with it, you can reposition it. But when you have a very good acquisition quality, you don't need to adjust it. It's very accurate. And on top of this, it gives you the crystalline lens wise, the CLR. It's the distance between the straight line connecting between the ARs and the anterior surface of the crystalline lens. So next is this picture. And when you look at this picture, as a surgeon, as an ophthalmologist, say, this is the distance, and you want to put a dot here, draw a straight line to the other scleral spur, and you say, this is the distance of scleral spur to scleral spur. But unfortunately, you know the refractive index of the cornea and, of course, the aqueous in the anterior chamber is different. So when you are drawing a straight line on this image, you will not get the good result. What the machine does from itself, and you can rely on it, is actually it takes this lens rise on a line that's a little bit curved, and it does it automatically. It corrects for the refractive index changes between the cornea and the aqueous in the anterior chamber. So don't go fooling around with the numbers. What you get on the machine is what you need. And where do you get it? Here you get numbers. The scleral spur to scleral spur measurements are in the diagram in the CSO machine. For these measurements, we used the equation which was published by Nakamura in American Journal of Ophthalmology in 2018. And that formula tells us the optimal ICL size is this number plus 0.71 times anterior chamber width plus 0.65 times the CLR. And for this example, we have these numbers, we calculate this, and we got a number of 12.62. In this case, we implanted a 12.6 millimeter diameter ICL in this eye. So also cost-effectively, we can just judge what we predicted and what it actually was. In this case, 487 micron volt in the center of the ICL. In these my number of cases, this is the distribution 1 month, 3 months, 6 months, and 1 year after surgery. And you see all of them are within normal limits. By introducing the ICL, the version the V4C in 2011, with the central hole in it, we experienced that we don't need to create a larger vault. From a vault of 100 micron to 500 microns is sufficient. Why? Because the aqueous is forced in between the crystalline lens and the ICL before it goes into the anterior chamber. And what did we see in this 9 years follow-up? We only had one patient with cataracts. So in the more than 3,000 cases, we only had one patient with cataracts, because it's very important that there is contact with the aqueous and the crystalline lens during that period of time. Whether the volt is 100 microns or 500 microns, that doesn't matter anymore. So we are targeting more and more towards a lower volt and towards a higher volt, which we did in the older days before the aquaport was introduced into market. This OCT also gives you a lot of information post-operatively. You can see here the aquaport nicely in the ICL. You can see the crystalline lens. You can see the iris plane. So it's very interesting to check this after surgery. It gives you the vault, and this is a hyperopic ICL afterwards. And in this case, a very interesting case, not so fortunate for the patient. But here you can see that the ICL was inverted, so it was inserted in the eye upside down. And it's when you're looking at it, sometimes when you're not an experienced surgeon, you can miss it. When you have this the OCT evaluation, you clearly see that the iris is pushed forward because of the upward placement of the haptics, and it pushes the iris forward. So we can't miss it when we have this OCT evaluation postoperatively. Another major thing in the MS39 from CSO, the OCT, is that you can have a dynamic evaluation of the ICL after surgery. And then bear in mind, look at this, when there's a constriction of the pupil, you get the ICL vault diminishes. We think that the vault, or most of us think that the vault of the ICL postoperatively is a fixed number. It is not so. And when the iris moves, the vault also moves. So bear it in mind, vault of the ICL is not a fixed number. It's a dynamic number. And this is in a lot of papers published in the literature lately. You can see that there is one number for the vaults, but it only states in a certain dynamic pupillary state. So bear in mind that the ICL dioptrical power is very important, because the vault of them, let's take an identical eye, and put in a -2 ICL, and you get a certain vault, let's say 250 microns. Put in that same eye a -10 ICL, you will not get the same vault. So the power of the ICL is very important to determine the vault. The anatomy of the eye is very important, but also where do you place the haptics? Do you place the haptics in the same position, in the sulcus, on the zonules? This makes a huge difference. So we can calculate with what you want, but you will not always get the same vault when putting in an ICL in an eye, even when doing the same appropriate measurement. So bear in mind, this method is more practical for ICL sizing than UBM, because you need a very good technician to perform that measurement. And you have to touch the eye of the patient, which is not pleasant. Use the equation for ICL sizing. We can predict the vault and select the appropriate ICL size. The OCT and clear segment OCT showed higher accuracy compared to UBM. The vault prediction was 72% within 0.80 millimeters and 95% within 0.36 millimeters, with these astonishing numbers. It is non-invasive. You can use it pre and post-operatively. And it gives you a complete anterior segment evaluation. It predicts the vault in a very predictable way. And you can evaluate post-operatively the dynamic relationship between the ICL, the iris plane, and the crystalline lens. I thank you very much for your kind attention.
Thanks so much, Eric. It's a very nice presentation, and sizing the ICL is very important. It's a very elite surgery and need to be ended in a very nice way, starting by sizing. Any problem in the sizing is going to have complications post-op.
Exactly.
You have a lot of questions, and I'm going to start with a nice question. How do you take into account the natural crystalline lens growth?
Well, actually, you know, the crystalline lens growth by 50 microns a year. It's you don't have to take that into account because I told you with the center flow, there is a natural flow in between the ICL and the crystalline lens. And even when the crystalline lens grows, still the aqueous is pushed in between that space. So it will lift the ICL in the upcoming years. So you don't have to be afraid.
Another tricky question. What about the difference in vaulting between the central and the peripheral ICL?
Well,
with high versus low powers, would you still consider 100 microvolt is safe?
Very good, and indeed a very tricky question, Ashraf. Well, we know from the previous models that even if the central vault can be large enough, when you have touch in the periphery, you get lens opacifications and later on cataracts. But with this lens, with the aquaflow from since the V4C, because of, I told you so, the aqueous is forced in between crystalline lens and the ICL, and it lifts the ICL. The weight of the ICL is very low, so the ICL will be lifted away from the crystalline lens, even in the periphery, in most cases. Even when the crystalline lens grows, this is not an issue. As I told you, in 9 years follow-up, I only had one patient with cataract formation, and we can discuss whether it is caused by the ICL or is it caused because the patient was a -12 myope. So this, I cannot answer the question, but just bear in mind it's only one case in more than 3,000 ICLs in pound.
How you explain the deposition of iris pigments? You find it in the first 2 weeks post-operative. It's an improper sizing, or it's iris shaving, or what?
Well, yeah, that's why the anterior segment OCT is so valuable in the pre-op evaluation. You have to look at the iris configuration, how is the iris sitting on top of the crystalline lens, and there you can also predict when you do surgery, when you put an ICL in the posterior chamber, which eye will get some iris shaving and then the pigment release. It's very important to take this into account before doing surgery. Preparation is the success after for having very good post-surgical results. So do a very good workup before surgery, do an anterior segment OCT before implanting a ICL.
From your 20 years experience, what is the instance of retinal detachment, glaucoma, and cataract with ICL?
Oh, yeah. Well, retinal detachment, the incidence in my population of retinal detachment, and you can also find it in the peer-reviewed literature, is not higher than the normal population. It's completely different than doing a refractive lens exchange when you don't have a posterior vitreous detachment, which is, we all know is much higher than in the normal myopic population. So with ICL surgery, I do not have any concern without a higher incidence of retinal detachment. Glaucoma as well, we take great care in looking at the angle, preoperative angle with the OCT. And we have a narrow angle. We advise the patient to not have surgery or to choose another type of surgery. So glaucoma is predominantly related to a narrow angle if you implant the ICL. So just take great care in looking into the preoperative angle before putting in the ICL.
Thank you so much, Eric. And we have a lot of questions, but at the end, we are going to answer more and more. Thank you so much.
Thank you very much, Ashraf. Have a nice Easter, and I was very happy to be part of this webinar.
Thank you. You stay with us. Still, we are on. Can you remove your presentation, Eric?
Yes, I will.
Okay, and now I take the ball, and I would like to present the soul of these meetings. Is Dr. Ashraf Armia, one of the world renowned anterior segment surgeon, and
Magul, please put on your unmute your.
Okay, could you hear me? Could you hear me?
Yes, yes.
Okay, I'm going to
muted.
Yeah, I'm going to present now the soul of this event, Dr. Ashraf Armia, that will talk about the toric ICL and toric IOL for keratoconus refraction correction. So please introduce.
Thank you, Miguel. Thank you. So I'm going to speak after three speakers. They are eminent in their specialty, but I'm going to take my talk from the end of each one. So I will never do a toric ICL or toric IOL for any keratoconic refraction unless it is a stable keratoconus. So if it's a stable, it needs a cross-linking or proper cross-linking. And I'm not going to use the ICL unless I do a proper sizing for these cases, because it's not a straightforward cases. Everything in the anterior chamber is not straight. So I'm going to start. It's two parts of my presentation. The first is the using the with a toric ICL. The implantation of a toric phakic IOLs in eyes with keratoconus is complex. There is a higher order abrasion due to the complicated interplay with the phakic IOLs toricity and the corneal irregularity in these patients. So only subconus patients will benefit truly from the phakic IOLs, and it's very important to choose the correct one. The toric Visian implantable columnar lens from Star Surgical Company can be used to treat high residual myopic astigmatism up to -20 diopter sphere and 6 diopter astigmatism, in a particular after treatment of a regular component of the astigmatism. Phakic IOL implantation is an advantage in such cases because it's a cornea sparing procedure. Phakic IOL implantation is reversible. If it doesn't go well, you can remove it, the lens explanation. Phakic toric ICL can correct the high degree of myopic astigmatism without inducing a new high order aberration. Where to do toric phakic IOLs fit in? Over 21 years of age, had a stable refraction for 1 year after cross-linking. It's a contact lens intolerant and significant improvement in best corrected distant visual acuity with refraction. It must be eligible patient, has no other ocular or general pathology, and no greater than stage 2 keratoconus. Additional inclusion criteria were clear central cornea, central or centralized with the ICR cone, and anterior chamber depth greater than 3 millimeters, and mesopic pupil size of less than 6.5 millimeters. These are the criteria. The lens power calculation was performed by online, as Eric said, and the calculation is based on the manifest refraction. What is the manifest refraction? It is the axis of astigmatism in the axis obtained by manifest refraction. Cornea curvature, cornea thickness, anterior chamber depth measured by the pentacam. As Eric said, we are doing the sizing, and the metropia is the post-operative target refraction. A minor clinical adjustment of the anterior chamber depth was performed by subtracting no more than 0.2 millimeters whenever corneal anterior bulging was advanced. In patients with a good best corrected visual acuity and high residual refractive error after cross-linking, toric ICL implantation provides adequate correction of the ametropy. Very important to know that flattening, which is achieved by the cross-linking, is not significant enough to alter the mean spherical equivalent manifest refraction at the time of the ICL implantation, or the outcome of the ICL procedure after 12 months follow-up. I am going to give you an example how we use to put the ICL in a video. And for anyone don't know how to load the toric ICL, it's like the normal ICL, but it has a toric. So you take it, you put a normal fluid in the cartilage, very easy to put the side. Then you press from the other side to make a bolt of the ICL. This is how you load it. You put it at the edge. Then you invert the cartilage. And with special forceps, you catch and you take the cartilage away, not the forceps. You are not traction from the forceps. And you make sure that the lens is already loaded in the perfect way. Then you mark your 180 degrees, and usually measure in the toric ICL. Usually it comes with where you are putting the axis of the toric ICL. So in this case, it was about 25. I put a viscoelastic, viscodispersive. I open in the axis where I'm going to put the ICL on. Make sure when you unfold the toric ICL in these cases, because the cornea is not easy, you can see well, but make sure that when you unfold it, it's not upside down, because this is a major problem. You remove I usually do, I put the 4 haptics, the first one and the other opposite one, to make the lens stable. Then I do the reverse, this one and the other side, so it's now well formed. I remove the viscoelastic, and very important to make sure where you put your toric ICL in the same axis. Very important to remove the viscoelastic, make hydration, and make sure before you leave like this, the axis of the ICL, the same axis you are already used with the ink. So my take-home message for the toric ICL implantation is an effective, predictable, and safe to correct refractive error and improve the visual acuity in patients with a stable keratoconus. An accurate ICL calculation is very challenging in moderate and advanced keratoconus. Refractive outcomes depend on the accurate manifest refraction and intraoperative accurate axis alignment measurement. So what about keratoconus with cataract? You cannot put an ICL, but you can put a toric IOL. As we all know that the keratoconus is a common ectatic, it's very observed that in these patients are develop lens opacities earlier compared to non-keratoconic patients. So the correction of both astigmatism and the cataract with the phacoemulsification and the implantation of toric IOL can be an effective, safe choice for these patients with cataract and keratoconus. However, predicting the refractive outcome in these cataract patients is difficult with keratoconus, and the surgeon should be aware of different sources of biometric errors and possible consequences. The selection of toric IOL based on the K reading from the Shinflu image or the IOL Master 700, which is the best now to do the calculation. Axial length may be measured by IOL Master with immersion ultrasound biometry in cases of very dense cataract. The Higgs formula is used as a standard formula for the myopics, and the Hofer Q formula for the eyes with short axis less than 22 millimeters. Implantation of a toric IOL after phacoemulsification because of cataract in patients with keratoconus has its pitfalls. Performing cataract surgery in patients with keratoconus needs planning and careful consideration. This is not a normal eye. Very important to know that keratoconus is a problematic and defined. You can misdiagnose it as a keratoconus or other things, not a keratoconus patients. Accurate keratometry measurements are difficult with the results of inaccurate cornea and difficulties in selecting the power, very important, selecting the power of the IOL. In eyes with keratoconus, it is not be assumed that the measurement of the K is equal to the K at the visual axis, nor that the effect of the measurement error is uniform in all keratometric values. Very important to know that in eyes with K-max is less than 55 or equal diopter, this effect is usually small, and using K values for the IOL Master results in acceptable refractive outcomes. But compared 5 different keratometry measurements from Pentacam or Biscan or Master Ice or Mayo or Gival, and also found good reliability of the K values up to 55 diopters with all devices. But if the K max is more than 55 or equal, all devices has low reliability. The difference between the planned refraction and the spherical equivalent, very important to know, it's defined as the biometric prediction error. Very important. So if the study showed that in mild keratoconus, which is defined as a K max is less or equal 48 diopters, the biometric prediction error was 0 diopters. And in moderate keratoconus, which is 48 to 55, the biometric prediction error was minus 0.3 diopters. But in severe keratoconus, which is above 55 diopters, the error was 6 plus 6.8 diopters when using actual K values, but only 0.6 when using the standard K values. Traditional thinking has been to avoid using the toric IOLs in keratoconic eyes because they could increase the eye already irregular astigmatism. But however, the availability of cross-linking topo guide, as mentioned by Miguel, allow more patients to receive toric IOLs at the time of cataract surgery. So we do this before the cataract surgery. The surgeon should explain to the patient that this is not an astigmatic debulking procedure, rather than one will correct all errors. The patient may still need glasses or contact lens for the best vision, but the outcomes can be very good. It's very important to watch the progression of keratoconus after cataract surgery. The patients who are happy with a hard contact lens or scleral lens, but cautious in placing the toric ICL, it's important to counsel the patient because not all the patients with a toric IOL will going to have the same vision with their satisfied hard contact lens. What's spherical abrasion? Spherical abrasion, when the correct IOL power is determined, the surgeon should be aware of the positive and negative sphericity of the IOL. This is a very important issue. So, in the keratoconic eyes is unique in its sphericity. More corneas increase the positive spherical abrasion over time. So, when we use the IOL, because most of the IOLs have a slight negative spherical abrasion, one should consider choosing true aspheric lens such as the E-Vista or the support from the Bosch and Lomb, which has a 0, or a positive spherical lens such as sensor from G&G. Refractive surprise, it is recommended to use an intraoperative handled autorefractometer to improve the refractive outcomes in these patients because refractive surprise is common in these patients. I have some intraoperative tips, which is very important in doing the cataract in these patients. Early keratoconus can be managed like a normal cataract, but severe keratoconus with steep medians and possible cornea scars can be troublesome. When the main incision is performed in cornea with abnormal structure properties at the keratoconic eyes, post-operative K values and corneal shape can change in unpredictable manner. Cataract main incision site should be planned during the preoperative examination. In cases of inferior temporal cone, the main incision should be placed superiorly or superior temporal. Vice versa, in the rare cases with a superior steep cornea, the main incision should be placed temporally. In the presence of corneal scarring, this is a big problem because it's common that the main incision we're going to place at 90 degrees apart from the corneal scar. In case of inferior scarring, the superior approach is not advisable because the phaco-tip will be under the scar and you are not going to see well. Also, the keratoconic corneas are thinner and floppular more than the other corneas. Therefore, clear corneal wounds are more prone to leak post-operative. So, you can take sutures. It's not a harm or it's a crime for these patients. Also, the corneal sutures, the well-constructed wound is very important in these cases. In the presence of diffuse scar, surgeon should expect the worst view for the patients. So you can tilt the microscope to see better, Also, there is, due to the different light sources, you can do some staining for the corneal capsule. Another trick, you can put a viscoelastic because it's going to spread over the cornea and remove the corneal irregularities and make some little magnification during your work. I'm going to show a small video about how you put in these cases. So this is like the normal cataract as usual. But in this case, very important that your rexus must be very central and complete and continuous, because you are going to put a toric IOL in a cornea is not normal cornea. So any tilt of the IOL or any decentered is going to have a high order aberration for the patient. So you are continuing as usual. As you do your phaco wish, very important to polish the posterior capsule perfectly and remove all the cortex and don't leave any cortex in the bag in these cases, because capsular phimosis in these cases can alter the vision for these patients. Very cautious to remove it, very good. And you are marking the 180 degrees from the start. Polishing again. And then you mark the manifest, where you are going to put your toric IOL. In this case, it was about 25 degrees. I'm using in this case, the thickness from J&J. It's very good because you have 4 dots instead of 3 dots. So if the pupil is a little bit closed, you can see the 4th dot very close. You should remove all the viscoelastic before under your IOL. And before removing out for hydration, you should hydrate your wound and make sure that, the axis of the IOL is the same axis where you are planning from the start. Hydrate your wound and make sure that the points in the same side of the marking. So my take-home message in these toric IOLs in keratoconus, we will continue to see more keratoconic patients in our cataract evaluation. The expectation of these patients can be just as high as those of other refractive cataract surgery patients. To obtain the best outcome of these patients, surgeon must perform proper patient counseling, be careful with post-operative measurements, adjust the refractive target based on the calculation made with the multiple formulas, and have a plan in place to address the refractive surprises with a stable, non-progressive keratoconus. Thank you so much. And after what I are going to face, we are going to welcome you again in our country, Egypt. Thank you for attention.
Thank you, Dr. Cosmo. Thank you, Maguel. Thanks for Eric. And thanks, Dr. Ashraf Armia, our eminent Egyptian international speaker, for what you have shared today. Really was perfect and great added value to all of us.
Okay, we are going to have some questions rapidly because yes, I have 2 questions. Wait a minute. One for yes. This is it was for Eric. I don't know, Eric is still with us?
No.
No. So it was done. Yes. This is for how can we choose the axis of the part? No, not you. Not you. Not you. Yes. This is for Miguel. Transepithelial PRK is a work in which the laser removes also the epithelium and model the stroma. Is this true?
Yes, the transepithelial PRK is a modality in which the laser removes in the same time epithelium and stroma. So the reason this is really an advantage, because you do not touch the patient, you do not use alcohol for epithelium removal, but more important, you exactly decide how much larger is the treatment and how much you can decenter on the cornea.
Another question was for Cosimo. Someone said that if we can remove the epithelium like the epi LASIK, can we do the procedure with it?
Yes, I used EBK system. You know the EBK?
Yes.
Yes, of course. I used this in the EPIOF procedures. Yes, it's possible.
Okay.
Of course.
And
it is very safe for the Baumann.
Okay. So
it was an interesting question. The choose of axis for the toric IOL. If you choose the axis based on topographic axis or aberrometry axis, what you choose for the axis of toric IOL?
I use it for. Yes, I use that. I compare with the manifest refraction with the topographic astigmatism. I usually use it both.
As you already told, Ashraf, corneal regularization in this patient really improve the biometry a lot. I had some experience with this patient. It's not a 2020 unaided surgery, this. So this is important to explain to the patient. And also, you're going to correct a defect that is the result not only of anterior surface, but is the result of posterior bulging. So anyway, I have a very, very beautiful experience in terms of improvement, but also spectacle and also contact lens is mandatory after for correct well this patient. But I saw a lot of improvement when I regularized before the corneal surface. So sometimes I perform a regularization also in older patients in which the cornea is very irregular, because we can change cornea before, not after.
Yes.
After is a mess.
It's a very yes. And there is a question: do you consider the in-situ torsion when marking the IOL axis? Yes, because I usually mark the patient before the local anesthesia on the slit lamp. It's the best way to mark the astigmatism before anything. So it's very nice. I'm happy that everyone is here, and I'm happy for this meeting. Thank you so much, Cosimo. Your presence is very very important for us. Thank you, Miguel, and thank Eric for sure. And thanks for Eva for taking the care of this meeting. Thank you so much.
Thank you all for your presence and for your time, for your effort, for your knowledge. Actually, knowledge is power, and you are so powerful. Thank you all.
Thank you, and happy Easter to all.
Happy Easter for everyone. Thank you. Thank you. Bye-bye.
Bye-bye.
Bye-bye.